Hi, everyone. Thank you guys so much for joining us this evening. Just to introduce myself a little bit, my name is Hissini Menta, and I'm a rising senior at California High School. And today I will be talking about probing the SAR of novel C-4 carbonate and carbamate podophyllotoxin analogs and photocages, tubulin inhibitors with antiproliferative activity in cell models. Okay. So just to begin, I'll be starting with introducing what podophyllotoxin is and then what its impact is, and I will further move into our lab's previous studies on the project and our current efforts. So to begin, podophyllotoxin is a natural product that is extracted from the wild mandrake plant that's pictured here on the top left corner. The wild mandrake plant was traditionally used by various indigenous groups for medicinal purposes. However, it still holds great importance through its ability to inhibit tubulin polymerization, which does prevent formation of the spindle fibers during the cell cycle. The inhibition of tubulin acts upon mitosis when spindle fibers pull away from homologous chromosomes that hinders the cell cycle. So through previous literature, we have found that over 60% of FDA-approved small molecule therapeutics consist of natural products and their derivatives. Knowing that, we can see that in this image on the left, that natural products and natural product-inspired derivatives outnumber natural products themselves in a 10-to-1 ratio. While evaluating that case that natural products are not optimized, probing a need towards the modified molecules. So creating these modifications of these natural products can be used to better target human disease. Okay. So here we can see visually the binding of podophyllotoxin to the alpha-beta interface to identify the mechanism of action. Our compound podophyllotoxin binds to tubulin, which inhibits and where the tubulin dimers polymerize to form molecules. Okay, so there have already been many modifications at the C4 position that have led to many FDA-approved and clinically tested tubulin inhibitors. As we can see here with these 8 different compounds, first and second, we can see etoposide and tenopposide. And furthering in on these two FDA small molecules of podophyllotoxin are modified just by adding a sugar at the C4 position, which leads to DNA topoisomerase inhibition, which varies from the actual parent compound mechanism, where podophyllotoxin actually inhibits the tubulin polymerization. So topoisomerase is an enzyme that is vital in DNA replication, and the inhibition leads to DNA cell death and DNA destruction. Through this, we can identify an importance at the C4 hydroxyl group, leading us to our further studies through the modification at the C4 position. To the left of this, we can also see the visual binding of etoposide to DNA topoisomerase. Okay, now I'm going to go over the previous work that our lab has done. So here we can see that in our previous study, this was also the interface between the modifications at the C4 position and cytotoxicity. As seen through our novel ester compound library in the bottom, 2A through 2F, we can see an increase in bulk at the C4 position and hydrophobicity. So previous studies in this area have not identified a clear trend in this relationship at the modification at the C4 position and its potency, but since this is unknown, our goal was to investigate how increasing bulk and sterics at this position would impact biological activity. So with these novel ester compounds that we can see from 4-acetate to 4-phenylacetate, we went towards testing biological activity through MTT assays, flow cytometry, tubulin polymerization assays, and computer modeling. So after all those assays and ex vivo assays, we were able to look at this and see that in these previous studies that identified the increasing bulk at the C4 position and hydrophobicity, we actually see a decrease in cytotoxicity that we were able to identify through our in vitro assays. However, with our flow cytometry experimentals, we were not able to identify a relationship between the C4 bulk and tubulin inhibition. So due to these incomplete conclusions that were drawn, we have expanded our library of compounds, including a library of carbonates and carbamates, to establish a more clear SAR. So here we can see the one-step synthetic route yielding these novel carbonate analogs. It's one step, as we can see above, and then we have these 7 analogs at the bottom. And then that was our carbonates. And then looking at our carbamate synthesis, we did have to screen a lot of different conditions due to its unknown synthetic route. And here we can identify the two-step synthesis, yielding us with our novel carbamate analogs. So with this synthesis in particular, we have the nitrophenyl carbonate, which is used as an intermediate. So with the addition of this amine after the nitrophenyl carbonate intermediate, we were able to identify instantaneous conversion due to the indication of a reaction turning yellow, which is a nitrophenyl leaving group. So the nitrophenyl leaving group is a good leaving group in particular, meaning is a good leaving group in particular. So when the amine was added right away, we were able to see the yellow change of color, and the amine was attached to the C4 position. So with a successful synthetic route that we were able to establish, we were able to synthesize a large library of these carbamates. And then first, we're looking at the alkyl carbamates, as we can see here, from methyl, isopropyl, isobutyl, T-butylhexyl, and then further on. And then we also were able to synthesize more cycloalkyl carbamates. And then finally, we had an addition of our heterocycloalkyl carbamates as well. Now we're going to move on to the preliminary biological work that we were able to do with our carbamate and carbonate analogs. So we did some in vitro and ex vitro biological studies conducted on our array of carbonate and carbamate analogs. So as we can see on the left, this is an MTT graph. So an MTT assay is basically a colorimetric assay that is used to measure the metabolic cellular activity. So basically a simplified procedure that we can kind of identify through MTT is it involves drugging cells with our compounds at various concentrations, ranging from 10 micromolar to 5 nanomolar, to track the percentage of cell viability. Basically, we're tracking how many cells we still find alive after different drugging concentrations. And on the left of this graph, on the right of this graph, we're able to look at the flow cytometry analysis, where our compounds are also drugged and analyzed, which is used to identify which stage of the cell cycle our compounds actually kill cells in. And seen through this graph, we can see that Taxol was used as our positive control at the top, and then we use our parent compound Podophyllotoxin as our normal and our baseline. So as we can see, all these compounds inhibit tubulin at pretty similar but varying extents. So since these analogs of Podophyllotoxin should inhibit tubulin, a protein that is present in the G2 through M phase, ideally, our compounds will kill during that phase. Okay, and again, moving on, we're able to see more of our analogs. This time we're screening all 29 of our analogs, including our ester analogs. So our compounds were preliminary tested on cell lines MCF7, MDA-MB468, and HT-29 cells, MCF7 cells, which are breast cancer cells, 468 cells, which are triple-negative breast cancer cells, which mean that they do not have the 3 cell markers, estrogen, progesterone, and a specific protein. And then finally, the HT-29 cells, which are human colorectal cell line. So looking at these two graphs, neither of this data was very evidently conclusive, as seen on the 468 cell line, where we have viabilities all around 20 to 30% within the 24-hour timeline. While we don't see a very great differentiation between our analogs, we do see a high compound potency. And looking at our HT-29 MTT data, we can see that overall, they do have a higher cell viability, but that we do see lower viabilities at the micromolar concentrations, but rather high viability when we move up to nanomolar. With these differences between these cell lines, we were not able to fully conclude any results, and we hope to test these further through replication and different cell lines. Okay, now we're moving on to 2 other factors that we use to evaluate our compounds using C-logP and TPSA. So C-logP, which is the graph on the left, we can see that the higher the C-logP indicates more hydrophobic compounds, which are more nonpolar and less potent. In relation to these TPSA values that we see on the right, we're able to evaluate cellular absorbance, and the lower the value, the more permeable the compound is in cells, which in turn increases the impact of the drug. So overall, higher C-logP values mean more hydrophobic, more nonpolar, and less potent. We're hoping to find, as we can see Podo being the lowest C-logP value, and then all the other analogs having higher C-logPs. Now, in relation to our carbonate and carbamate project, we do have a lot of future work that we will be continuing over the next couple months. So we will start by resynthesizing and repurifying any of our carbamates and carbonates for our in vitro and ex vitro assays. So with our 24 analogs that we do have, we do need to test for purity through NMR, and based on results, we will determine which compounds we will need to resynthesize and which we need to repurify in preparation for these assays. We will also be performing a lot more MTT cytotoxicity assays on cell lines 231P, 468, MCF7, HT-116, HT-29, CalU1, and HEC-293. And these cell lines contain an array of breast cancer, triple-negative breast cancer, colon, colorectal, lung, and kidney cell lines. Once we have gathered sufficient MTT data in hand, we plan to calculate the IC50 values for these analogs. So IC50 is a quantitative measurement that we use that is used to determine the concentration of a drug where 50% of the cells die. So a higher IC50 value that indicates a lower potency in your compound, and more is needed to kill the same proportion of cells. The lower the IC50 value, the less compound needed and the more toxic your drugs are. We will also be continuing flow cytometry analysis to track for cell cycle inhibition, and we will also move on to tubulin polymerization assays, which are cell-free. So now I'm going to be moving on to a more controlled and localized release of our Podophyllotoxin constituents of our photocage compounds. So overall, some of the biggest challenges that currently are seen in drug discovery are being able to modulate the active concentration of these drugs when they are released. So as we can see in the graph in the top left corner, we can identify two curves. The curve on the left showing the therapeutic window, and then the curve on the right is showing the toxicity. So as we can see, the one on the left shows when not a high enough potency or amount of drug is given, we do not see, we see that not enough cells die. And then when we move on to the toxicity, moving on to the curve onto the right, cell death is more prominent. However, it's not selective. So we do see this box in between these two curves known as a therapeutic index. And this is actually the ideal therapeutic window. And achieving this window is very hard without a means of controlling the delivery mechanism. So as I mentioned earlier, our compounds are very potent, and they do exhibit a broad spectrum of antiproliferative activity. However, we want it to be more selective. Also seen in the image in the left bottom corner, we can see the controlled localization and the importance of that in modulation. So the strategy that we are optimizing to combat the selectivity is through the prodrug route. So a prodrug is basically something that consists of something that is chemically ligated to it. So over the years, prodrugs have actually been used to respond to different factors such as pH, enzymatic hydrolysis, and redox events. However, our focus actually looks into light. So we look to use photons for a light triggering release, and with the right wavelength and quantity. This is where our approach to photocages come into play. So a quick overview of our kind of synthetic pathway for photoreleasable photocages that use a photolabile nitrobenzyl photocages in for this. So our first synthesis with our starting material is the 2-nitrobenzyl alcohol, and the second starting material would be the 4,5-dimethoxy-2-nitrobenzyl. So we were able to form two different carbonates, the 2-nitrobenzyl and the 4,5-dimethoxy carbonate. And once we were able to make these original chloroformates, they would be attached to podophyllotoxin, where we're able to make the final photocage compounds. So here also we can look onto the right side, and we see two different graphs, and these show the concentrations of the two photoreleased carbonates over time. So as we can see, the graph on top, the 2-nitrobenzyl, as we track over time, over a course of 17 minutes, the concentration of podophyllotoxin does increase as light is shown upon it, as we can see on top when UV radiation is shined upon. And then when we look at the graph on the bottom, which is a 4,5-dimethoxybenzoyl carbonate, again, the more time that goes by, and then we see a higher concentration of podophyllotoxin being released. Now a little quick overview through our LCMS data that we were using to validate our photo release compounds. So we can see in the first two graphs that there is no peak in these two circled areas where podophyllotoxin should actually be seen. And that means that no photo release is happening because these two are not being exposed to the UV light. However, when we look at the bottom where we see the 2-nitrobenzoyl carbonate that has been radiated with UV light, we do see a podophyllotoxin peak. So that shows that after UV, we can clearly distinguish the podophyllotoxin peak that is activated by the light, which is not seen in the other two. Okay, and then our last kind of way that we were able to kind of look into our toxicity and how these compounds work is we did more cell assays. And here we can see two other graphs. We can see the two compounds in dose-dependent MTT assays. And again, as time goes over, cell viability does decrease over this 48-hour period in different concentrations. So we do see lower viability in cells over the course of 48 hours, but it's not very prominent. And then overall, our photocage compounds basically started with the idea that there aren't a lot of derivatives of podophyllotoxin that have the ability to arrest the cell cycle. And with this low specificity, we know we have to take an alternate approach to kind of control how the drug is acted upon. So the method we did use was prodrugs, which are biologically inactive, and they are activated through certain conditions. And so this led to our targeted approach towards the drug release, as well as the overall reduction in the drug's toxicity, but not the potency. Thank you guys so much for listening. That is the end of my presentation. I would just like to thank my advisor, Dr. New, and the group members.